In this video, we're going to take a look at proteins. Proteins have many functions, and there are many examples of proteins, including antibodies such as IgM and IgY of the immune system, enzymes such as trypsin and pepsin that take part in many internal reactions, structural proteins such as collagen that are the most abundant type of protein in mammals, as well as transport proteins such as hemoglobin that partake in the exchange of oxygen and carbon dioxide. We're going to encounter many such proteins along your journey in molecules and cells. But before we move on, let's review some basics. What makes a protein? A protein is a polymer that is made up of a sequence of amino acids. In this case here, each orange box represents an amino acid. Each amino acid consists of a central carbon to which is attached a carboxylic group, an amine group, a hydrogen atom, and an R group that represents an organic substituent. It is the variation in the R group that gives rise to the 20 different amino acids in humans. So how do amino acids come together? They come together through a reaction known as a condensation reaction that utilizes energy. In this slide here, we have two amino acids. They come together using energy in the form of ATP to form a peptide bond. This reaction is a condensation reaction, and that's why water is released in this reaction. Notice that the peptide bond is between the carboxylic group on one amino acid and the amine group on another amino acid. The R groups do not partake in this reaction. In the opening slide of this presentation, I showed you a few of the thousands of different forms and functions of proteins, including enzymes and antibodies and hemoglobin and collagen. It's the different combinations of sequences of amino acids in the protein that leads to its final shape and function, and this is what this video will focus on. There are four main levels to protein structure. The primary level represents a sequence of amino acids starting from the amino end to the carboxylic end. In this figure, I'd like to highlight the amino end, which is the amine group on the first amino acid, and the carboxylic end, that is the carboxylic group on the last amino acid. These amino acids, indicated by R1, R2, R3, and R4, are connected by peptide bonds, as you can see along the dotted lines. It's actually the peptide bonds between amino acids that are crucial to formation of the primary structure. The secondary level of protein structure represents two main structures, known as the alpha helix, pictured here on the left, and the beta pleated sheet. While peptide bonds are crucial for the primary structure, these secondary structures depend on hydrogen bonds, and that's between the hydrogen atom and an electronegative atom, which in this case is oxygen on the carboxylic groups. In the case of alpha helix structures, these hydrogen bonds are considered local, as they are on relatively adjacent amino acids in the helical structure. They're approximately 4 amino acids away. In the beta pleated sheet, however, these bonds are not considered local as they're farther apart. The beta pleated sheet actually has two different orientations. They can be parallel or anti-parallel. As you can see here, in the parallel case, the sheets are aligned in the same way, starting from the amino end to the carboxylic end. Whereas in the anti-parallel case, they're aligned in alternating ways. One thing to note between the parallel and the anti-parallel case is that the hydrogen bond is weaker in the parallel beta pleated sheets. This has to do with a slight bending of the hydrogen bond in the case of parallel beta pleated sheets that leads to a weaker bond. Before we move to tertiary structures of proteins, let's take a close look at motifs that are actually super-secondary structures. These usually have some particular function and consist of combinations of secondary structures. For instance, the helix-loop helix comprises two alpha helices that are looped together. So in this case, the two alpha helices loop together to form a binding site for calcium ions. Another example is the zinc finger that consists of an anti-parallel beta sheet and an alpha helix that aids in RNA and DNA preparation. Alright, so now we can begin seeing some functions appear in proteins. This is the perfect transition to the tertiary level of protein structure. This is the level which helps maintain the 3D structure of the protein and is associated with functionality. The tertiary structure consists of domains. Domains are actually combinations of several motifs, and several domains come together to form the tertiary level of protein structure. This level of protein structure is characterized by non-covalent bonds. Just to briefly recap, in the primary structure we looked at peptide bonds as being responsible, hydrogen bonds were responsible in the secondary structure, and in this structure we look at non-covalent bonds. A brief review on non-covalent bonds that are involved in protein structure are ionic bonds between charged elements, van der Waals bonds between and within the molecules, hydrogen bonds similar to the ones in the secondary structure, and finally hydrophobic interactions between nonpolar molecules due to their phobicity of water. Disulfide bridges or sulfur bonds are commonly found when describing the tertiary level of protein structure. These are actually covalent bonds and they function to help reinforce the 3D structure of protein folding. These are between the cysteine side chains in an amino acid. This is just a brief review of the tertiary level of protein structure. These slides are from Dr. Haas's lecture. In the slide on the left, you can see ionic bonds, van der Waals, and hydrogen bonds. These ones here show you the hydrophobic interaction. Notice how the green nonpolar side chains aggregate to exhibit hydrophobic interactions. And finally, we have the cysteine side chain over here, and you form the disulfide bridges. Alright, so we've discussed 3 levels of protein structure. The final level of protein structure, known as the quaternary level of protein structure, is formed by a combination of multiple peptides. 2 common examples of quaternary structures are antibodies. These consist of 4 peptides, 2 heavy chains and 2 light chains. And we have hemoglobin, which is comprised of 4 polypeptide chains, 2 alpha peptide chains and 2 beta peptide chains. And so, in conclusion, with different levels of folding, proteins are formed. Here's the primary level that consists of the sequence of amino acids, the secondary level that comprises alpha helices and beta pleated sheets. Motifs and domains find themselves between secondary and tertiary levels. The tertiary level is the final combination of domains. And finally, we have the quaternary structure that's made up of multiple tertiary peptides.